Welcome to the world of fungi. When you hear the word fungus, you probably think of an edible mushroom or bread mold. But fungi are so much more than that. You're wrong if you think blue whale is the largest organism on planet Earth. Actually, it is an amazing fungus spreading 2,385 acres, almost 4 square miles. The world of fungi is vast, interesting, miraculous, exclusive, unique, and functionally important. Fungi are usually multicellular, eukaryotic organisms that cannot synthesize their own food and feed by absorption rather than ingestion. They have diverse applications ranging from the production of enzymes and antibiotics to the bioremediation of xenobiotic compounds. Importantly, fungi are nutrient recyclers, environmental indicators, source of natural dyes and pharmaceutical compounds, act as plant growth promoter, and also utilized as biocontrol agents against a variety of plant pathogens. The fungal diversity can be appreciated by the fact that fungi neither belong to plant kingdom nor animal kingdom. Fungi have a kingdom of their own. Fungal diversity is second only to the insects in number of species. They are 6 times more diverse than plants. Only 5 to 10% is known. Thus, 90 to 95% is yet to be discovered. The fungal diversity research started at the Forest Research Institute, Dehradun, under the leadership of Dr. K.D. Bagchi in the Forest Mycology section under Botany branch and dates back to 1927. Later, this section was upgraded to Forest Pathology branch and was headed by Dr. B.K. Bakshi till 1977. Till date, significant contributions by different scientists have been made in this field, generating one of the largest fungaria and forest pathology museum, which contains nearly 12,000 specimens of forest diseases and fungi. One of the oldest specimens of Peridermium brevius on Pinus wallichiana needles dates back to 1877. Pictorial representation of different fungi in the form of handmade paintings at the museum had received recognition in the 8th International Exhibition of Botanical Art and Illustrations held at Hunt Institute, Pennsylvania in the year 1996. Similarly, National Type Culture Collection (NTCC) of FRI Dehradun harbors 988 live fungal cultures belonging to 140 genera and 298 species. 443 cultures of white rot fungi belonging to 57 genera and 115 species and 148 cultures of brown rot fungi belonging to 21 genera and 32 species are maintained at the NTCC. Important pathogenic, saprophytic, or medicinally important fungi are also part of this unique culture collection. These national assets are knowledge treasures representing biodiversity of Indian forest fungi. In recent years, DNA fingerprinting and sequencing has catalyzed a sheer expansion of mycological knowledge. DNA sequence analysis facilitates the establishment of taxonomic relationships among organisms on a firmer basis. At the Forest Pathology discipline of FRI Dehradun, molecular diversity studies have been conducted on important forest pathogens and medicinal mushrooms. The discipline also worked on the biodiversity of Ganoderma species and first time reported Ganoderma resinaceum and G. webrianum from North India, based on DNA sequence analysis. The species diversity of Trichoderma was also explored by DNA sequence analysis. The identified species have diverse applications such as enzymes and antibiotics production, bioremediation of xenobiotic compounds, plant growth promotion, biological control of plant pathogenic fungi and nematodes, and inducer of systemic resistance in plants. The largest fungal fruit body from India, which is enlisted as the world's third largest by Wikipedia, was discovered in the year 2017. Diversity of wood-decaying fungi of the northwest Himalayas has also been explored. The biodiversity of macrofungi on living trees, fallen logs, along roadside and grasslands has been explored in the New Forest campus of FRI Dehradun. In total, 187 species have been recorded till date. Work on the diversity of Felinus, a major heart rot pathogen, has been done. Apart from being used in bioremediation, some of the Felinus species, collectively known as Fansomba in Maharashtra, have medicinal value as well. Unfortunately, the habitat destruction due to anthropogenic activities and mainly on account of climate change has either caused shifting of the fungal habitats or their extinction. Therefore, red listing of fungal species has been given priority in many countries. In view of this, preparing a red list of Indian fungi using IUCN guidelines is presently underway, which is a necessity to impress upon the policymakers and conservationists. This initiative will attract, educate, engage, and facilitate activities by both professional and amateur mycologists for fungal conservation in India. Fungi regulate the plant diversity by preventing the dominating species and enabling a wider range of species to flourish. According to the ancient Hindu scriptures, Lord Brahma, Vishnu, and Mahesh have been regarded as the creator, preserver, and destroyer. Fungi on the planet Earth has the same role to play, and without them, life will cease to exist.